Today's lecture, we will talk about the amino acid chemistry. Amino acids are forming the basic unit that forms proteins in our body. What are the importance of proteins in our body? They have many functions in our body. For example, they have a transport function like the hemoglobin that transport oxygen in our blood, albumin that acts as a transporter for many drugs and for many substances in the blood. Also, they have some storage functions. The storage function example is the ferritin, which is found inside the liver. Also, they may act as an antibodies and help in the immune response. They may act as receptors like the stimuli receptors in the neuron. They are very important for the cellular function through multi-protein machines. They are important for the cellular structure such as the collagen that's present in the skin. It is a type of protein. They may act as channels that control the movement of the contents in and outside the cells. Moreover, they may act as enzymes that catalyze many biochemical reactions in our bodies. What is the biological value of proteins? Proteins can be classified according to the biological value into high biological value proteins and low biological value proteins. And by high biological value proteins, we mean proteins that contain all the essential amino acids. They are digestible proteins and contain all the essential amino acids. And most of the animal proteins, like the milk, meats, and eggs, are proteins of high biological value. On the other hand, the proteins that are not digestible or they are deficient in one or more of the essential amino acids, they are called proteins of low biological value. And these are mostly the plant proteins. عشان كده الناس اللي vegetarian that do not eat at all animal proteins, they must combine more than one source of plant proteins عشان to make sure that they can receive all the essential amino acids. Proteins are polymers of amino acids, and by this we mean that the amino acids combine together to form polypeptides, and then the polypeptides combine together to form proteins. Amino acids form proteins inside the ribosomes. This is the general structure of any amino acid. Any amino acid is composed of an alpha carbon or an chiral carbon that has an amino group, a carboxylic group, and a side chain. The change in this side chain is what gives us the different amino acids. So any amino acid, it must have a chiral carbon, an amino group, a carboxylic group, and a side chain. Most of the amino acids, the naturally occurring amino acids, are found in the L form. We know that they are stereoisomers or enantiomers. Amino acids may be found in the D form or the L form. Most of the amino acids found in our body are found in the L form. And this sketch represents a 3D structure of the amino acids. As we said before, it's composed of a chiral carbon, carboxylic group, amino group, and any side chain. Amino acids may be classified according to one of four criteria. They may be classified according to the structure of the side chain into aliphatic or aromatic amino acids, into the polarity of the side chain, according to the nutritional requirements, whether they are essential or non-essential, according to the metabolic fate, whether they are glucogenic, ketogenic, or mixed amino acid. Firstly, we will talk about the classification of amino acids according to their side chain. According to the side chain, amino acids may be classified into aliphatic amino acids and aromatic amino acids. The aliphatic amino acids may be classified as simple amino acids that they have a simple chain like glycine and alanine. Branched amino acid, their chain is a bit branched like valine, leucine, and isoleucine. Also, we have hydroxyl or sulfur containing amino acids. They have an OH group like serine and threonine, sulfur group like cysteine and methionine. They are acidic and basic amino acids. The basic amino acids, they have an extra amino group like lysine, arginine, and histidine. Acidic amino acid, they have an extra carboxylic group like aspartic and glutamic acid. Also, there are amidic amino acids, and they are derived from the acidic amino acids. When the acidic amino acids undergo a condensation reactions means the reaction between a carboxylic acid and any amine or any compound that has amino group like ammonia in this case. بيحصل removal للwater H from the amine and OH from the carboxylic group, and they form an amide bond. Okay, and water elevates. This is called a condensation reaction. The condensation reaction of acidic amino acids give amidic amino acid, like the aspartic acid. When it forms a peptide bond, it forms aspargine. Glutamic acid, it forms glutamine. Aromatic amino acids are amino acids that has an aromatic group in their side chains, such as phenylalanine, tyrosine, tryptophan, and proline. Also, there's what is called derived amino acids. Derived amino acids are amino acids that are usually derived from other amino acids. For example, the hydroxyproline, it's derived from proline by adding a hydroxyl group. And hydroxylysine, it's derived from the lysine. And these are derived amino acids that are found in proteins. يعني they may be found in the protein chain. There are other derived amino acids that are not found in proteins. They are found as sole amino acid, such as citrulline, which is found in watermelon, for example, and ornithine. Also, there is a non-alpha amino acids. Non-alpha amino acids, it means that the carbon is not an alpha carbon, such as the GABA اللي هي الجاما aminobutyric acid. It's a neurotransmitter in the brain. Beta alanine, the carbon is a beta, is not alpha, like beta alanine is found in coenzyme A. And taurine, it's found in bile, اللي هو موجود كمان في الريد بول التاورين ده. This is a classification كده for most of the amino acids. الاليفاتيك amino acids قلنا الالانين and glycine هما have a simple chain. في اليوسين، برولين، فالين. aromatic زي الفينايل، الانين، التريبتوفان، التيروزين. Also الاسيدك amino acids اللي هما الاسبارتيك والاسيدك، والاميدك بتوعهم اللي هما الاسبارجين والجلوتامين. sulfur containing cysteine and methionine. hydroxylic serine and threonine. البيزك arginine, histidine, and lysine. Okay, classification of amino acids according to their polarity. Classification of amino acids according to polarity into polar and non-polar amino acids. The polar amino acids are the acidic amino acids اللي هما aspartic acid and glutamic acid. The basic ones are arginine, lysine, and histidine. Also, the amino acids that have hydroxylic group or SH group, such as serine, cysteine, threonine, tyrosine. Also the amides of the acidic one اللي هما الاسبارجين and glutamine. All the other amino acids are non-polar. يبقى تاني الpolar amino acids هما الacidic and their amides. البasic amino acids الامينو acids الهيدروكسيل amino acids أو السلفر containing amino acid except الميثيونين. الميثيونين السلفر بتاعته is present as an ether group, ف it's not polar. يبقى the only group the sulfur amino acid that's not polar هو الميثيونين. Classification of amino acids according to their nutritional requirements. We will classify them to essential amino acids and non-essential amino acids. The essential amino acids are the amino acids that cannot be synthesized inside the body, and we must have them in our diets. While the non-essential ones, they are formed from other amino acids, there's no need to take them in the diet. The essential amino acids, we can memorize them by this sentence. Try this VIP mole. Try for tryptophan, this for threonine and histidine, V valine, isoleucine, phenylalanine, methionine, arginine, leucine, and lysine. This sentence, try this VIP mole, represents the initials of the essential amino acids. At last, we will classify the amino acids according to the metabolic fate. According to the metabolic fate يعني بعد ما بيحصل ميتابوليزم للامينو acids بتحولوا لايه؟ فبنقسم الامينو acids into ketogenic that will be a precursor for ketone bodies, glucogenic that can be a precursor for glucose through gluconeogenesis, or mixed amino acids that can form both, can act both as ketogenic or glucogenic. The ketogenic ones اللي هم الليوسين and isoleucine, they can be converted into acetyl-CoA and acetoacetate. دول ketone bodies. They act as a fuel for the brain and very important كمان for the formation of the myelin sheath. The glucogenic, they can form glucose through gluconeogenesis. Amino acids can be converted to pyruvate, alpha-ketoglutarate, 6-phenyl-CoA, fumarate, oxaloacetate, and all these through gluconeogenesis and other metabolic reactions can be converted into glucose. Mixed amino acids اللي هم lysine, isoleucine, phenylalanine, tyrosine, and tryptophan. Now we will discuss some properties of amino acids. We will discuss some physical properties and chemical properties. First, the physical properties of amino acids. What is responsible for the physical properties of the amino acids is the multiple charged groups present in the amino acids. زي ما اتفقنا الامينو acid بيبقى فيها carboxylic group وamino group. Mostly they are charged. وبالتالي الامينو acids are mostly polar compounds. Since they are polar compounds, they are insoluble in nonpolar or organic solvents such as benzene, hexane, or ether. And they are readily solvated in polar solvents such as water. Also, they can be precipitated from the aqueous solution through alcohol, لأن most probably الألكول بيعملهم denaturation. They are of high melting point عشان بيبقى عندهم highly stabilized crystal lattice. Their taste, some of which are sweet or tasteless or bitter. Some are used as flavoring agents such as the sodium glutamate. Some are used as artificial sweeteners like the aspartame. بنلاقي some diet sugars بتتكون من aspartame. The artificial sweetener جاي من aspartic acid and phenylalanine. Very important physical property of the amino acid that has many applications is the ionization of the amino acid. As discussed before, the amino acid, it has an amino group and carboxylic group. If both are in ionized form, the total charge here on the amino acid is 0. At this point, this ion is called as Witter ion, and the pH where the total charge of the amino acid is 0 is called the isoelectric point. يبقى the pH where the net charge on the amino acid is 0 and it exists as the Witter ion, it is called the isoelectric point. If the amino acid is present in an acidic pH, there is a protons in the media, the carboxylic group will accept the protons, and hence the amino acid will have a total charge, positive charge, and it will be cationic. The maximum ionization that can happen is called a P PK1. يبقى the PK1 is the pH at which maximum ionization happens, and it exists as 50% cation and 50% zwitterion. On the other hand, لو أنا حطيت الأمينو acids in an alkaline أو basic conditions, what happens that the amino group will lose the proton and it exists, the total charge will be negative and exists in anionic form. The pH where the amino acid exists as 50% anionic and 50% zwitterion, it is called PK2. The isoelectric point is the average of PK1 and PK2 if I like to calculate it. يبقى isoelectric point is the point that the amino acid will carry no net charge. All the groups are ionized, so they cancel each other. Hence, since they have no charge, they have no mobility in the electric field, they have no solubility, they will precipitate, and their buffering capacity will be minimum. If a solution, if we add HCl drop by drop, يعني acidic solution, the pH at which 50% exists as cation and 50% as a zwitterion, this is PK1. If more HCl is added, more molecules become cationic and the solubility increase. On the other hand, if we put sodium hydroxide, يعني basic solution, it will be anionic. The pH here is called PK2. So the isoelectric point is the average between PK1 and PK2. It's easy for the monoacidic or monobasic acids, but if the amino acid has two basic or two acidic group, the calculation of the isoelectric point here will differ. We will take the average of the pK of the two acidic groups في حالة الأسيديك amino acids أو the two basic groups في حالة البasic amino acids, and this will be the isoelectric point. يبقى في حالة الأسيديك amino acid, for example, we'll take average of PK1 اللي هي ال PK of the first acidic group and PK3 اللي هي ال PK of the second acidic group. We'll take average, and this is the isoelectric point. A very important application for the isoelectric point is the buffering capacity of the plasma proteins and hemoglobin. At physiological pH of the blood, pH 7.4, the plasma proteins and hemoglobin resists the change of the pH, and this is called the buffering capacity. And this buffering capacity is due to the histidine amino acids. الهستidine is a basic amino acids بيعمل ionization of imidazole group. The point of this amino acids إن البفرنج action بتاعه أو الزفيتراين بتاعته exists between 6 to 8 اللي هي almost the pH of the blood, and that's why it is the most important amino acid that acts as a buffer in the blood. If it's a basic amino acid, if we want to calculate the isoelectric point, it will be the pK of the first basic group, which is 6, and the pK of the second basic group, which is 9.17. So we will take average, it will be 7.6, and this is the isoelectric point of histidine, which is almost the pH of the blood, and that's why it acts as a buffer. Now we will discuss the chemical properties of amino acids. The chemical properties of amino acids, first we will talk about an anhydrine reaction. It's the reaction of alpha amino acids only for the amino acids that have an alpha carbon. With an anhydrine reagent, it forms a purple compound. It can act as a quantitative or qualitative reaction. Reaction decarboxyl group. First is the decarboxylation. Decarboxylation means the removal of the carboxylic group. For example, and it evolves as carbon dioxide. For example, the histidine forms histamine, tyrosine forms tyramine, tryptophan tryptamine, glutamic acid form gamma aminobutyric acids or GABA. It's an important neurotransmitter inside the brain. A second important reaction is the amide formation. The amide formation happens for the dicarboxylic acids. It means that for the carboxylic group in the side chain. It combines with an amino group or ammonia to form an amide, like the aspartic acid forming the aspargine, the glutamic acid form the glutamine, and this is a method for the formation of the non-essential amino acids like aspargine and glutamine. Also, an important reaction is the peptide bond formation. Peptide bond formation forms by the reaction of carboxylic group of one amino acid and the amino group of another amino acids with the removal of water, OH, from the carboxylic group and an H group from the amino group. It forms a peptide bond. And by the formation of the peptide bond or the amide bond, like if two amino acids, this is called a dipeptide. If 3 amino acids, it's called a tripeptide. More than 3 amino acids, it's called polypeptide. Also, there will always be an N-terminal of the polypeptide and a C-terminal of the polypeptide. Third, we'll talk about the reactions due to the amino groups. We have transamination. Transamination is an important reaction for the synthesis of non-essential amino acids. For example, we have one amino acid. It has an amino group. Transamination means we take the amino group from the amino acid and give it to a keto acids, and take the keto group of the keto acid and give it to the amino acid. So the amino acids that got the keto acid, it form a new keto acid, and the old keto acid that receives the amino group, it produces a new amino acid. This reaction takes place inside the body. So the amino acid that is produced is considered a non-essential amino acid because it can be formed inside the body. Another important reaction is the oxidative deamination. Oxidative deamination, it happens by the removal of the amino group and the addition of a keto group. Okay, so the glutamate, here it's an amino acid. If we remove the amino group and we added a ketone group, it's formed an alpha ketoglutarate, and this reaction is very important in the carbohydrate metabolism, as will be discussed later. Also, a very important reaction for the amino group of amino acid is the formation of the carbamino compound. This reaction is very important for the transportation of carbon dioxide from the tissues to the lungs through the hemoglobin, where the amino group of the hemoglobin proteins receive the carbon dioxide and form carbamino hemoglobin, which is able to carry the carbon dioxide to the lungs. Also, we'll discuss reactions due to the side chain. This reaction will be discussed in details in the practical, so we'll go through it fast. First is the transmethylation. Transmethylation is also important for the formation of non-essential proteins, non-essential amino acids or derived amino acids. For example, methionine, it released its methyl group to an acceptor and convert it to homocysteine. So this reaction is important in the formation of production of homocysteine. Also, ester formation through the OH groups, the hydroxyl groups amino acids, they form esters with the phosphoric acid, and this is very important for the formation of phosphoproteins. The reaction of SH group, like two cysteine amino acids, they are combined through their SH group, forming cysteine, which is an important compound inside the body. Phenolic groups like the amino acid tyrosine. Tyrosine has a phenolic group, it gives red color with Millen's reagent. Guanidine group that is present in arginine amino acid, it gives red color with the alpha naphthyl and sodium hypochlorite. Cysteine, it has a free SH group, it gives a red color with the sodium nitroprusside and ammonia. التريبتوفان. it has an indole group which gives a red-violet color with the dimethyl aminobenzaldehyde in sulfuric acid media. It forms a violet ring, and this test will be discussed in details in the practical. At last, we'll discuss some examples of naturally occurring peptide. Firstly, we'll talk about the glutathione. Glutathione is very important antioxidant inside the body. It's a tripeptide. Tripeptide means that it's formed of 3 amino acids combined together by 2 peptide bonds, which are glutamic acid, cysteine, and glycine. 2 molecules of glutathione can combine together through the disulfide bond, form oxidized glutathione, and this is the oxidized form of the glutathione. Another example of a naturally occurring peptide is the adrenocorticotropic hormone. It is composed of 39 amino acids. Also, the bradykinin, it's a hypotensive agent that is released in the body. Some antibiotics like the gramicidin, it contains phenylalanine. Also, the thyrotropin releasing hormone, it is the hormone that is responsible for the release of the thyroid hormone. It's a tripeptide بيتكون من 3 amino acids which are glutamic acid, histidine, and proline. Thank you.